All right, cool. Let's get started. So, we will start with our weekly check-in. Nice little way for us to build relationships and to have a little fun before we dive into the content. So, let's have that little put it off. Cool. So, I'm going to randomly pick someone to share. The first thing we'll do is our random question, which is about ranking the meals from best to worst, breakfast, lunch, and dinner. Person answering that question today is going to be Tiana. The best is dinner? And then next is breakfast? And then lunch is last. Why did you put lunch last?
I didn't eat lunch.
You don't even eat lunch. That's a fair reason. I know a lot of people put breakfast last because they don't have a chance to eat breakfast. How many put that in the same order? Gentlemen, mask up. There we go. Cool. Shall I pick someone else just so we can see if they have the same choices? Klein.
I got one.
Huh? Same one. So you had dinner, breakfast, lunch. Did anyone put breakfast last? In my other class, a lot of people put breakfast last because they're like, I don't have time to eat breakfast. I don't really care for breakfast. Did everyone put dinner first?
Yes.
What did you put, Charlotte?
Breakfast.
Breakfast first. Interesting. Not a lot of people like breakfast. Why did you put breakfast first?
Because it's the only meal I eat.
That's the only meal you eat, don't you? You guys need to make sure you're getting 3 meals in, please. And the lunches are given to you free by the school.
Yeah, I don't like lunch.
Picky. All right. Cool. Anything else that we want to chit chat about for breakfast, lunch, and dinner? Shall we move on? Moving on. All right. Next one is the micro goal. So the person sharing that today is going to be Vatohi. What's your goal for this week?
Finish my health assignment.
Finish your health assignment. Yeah, I saw that on a lot of the weekly check-ins from my mentor class as well, that there's a health assessment due this week. So I understand if you guys have your focus on something else, and I understand if you're not going to complete a lot of science work this week for that reason. That's understandable. You guys got to prioritize. All right. Anybody else have anything they'd like to share? No? All right. Cool. Let us switch gears. Let us put away the phones so we're nice and focused, because I find them quite distracting. All right, so we are thinking about what we are learning this week on our little learning journey. So to recap, yes? There's a note, that's weird. I haven't gotten a note in a long time. Okay. All right, so to revise what we did last week, we started looking at that genetic variation and we did some revision for that. Is that again? Yeah, what's up? Yeah, is that a question? So we did some revision on genetic variation that you did in that remote learning. Yeah, I don't know. Okay. If you guys remember, I'm going to wait till you guys are done. This is really disruptive. It's not fair for us in class. Right? So we recapped some of the remote learning that we did with the genetic variation. Remember, we did put a pause on that external to 1, give you guys a chance just to play some of your own catch-up, and 2, because everyone was all over the show, it was kind of very hard for us to teach it. So use this time and also the holidays to play a little catch-up. We will pick it up. But I have a feeling we won't pick up it until maybe term 3, so you do have a while, but I'll double check with Ms. Quigley. So we started then our next topic, which is going to be an internal, and that's going to be on biological investigations. So since we're talking about investigations, we revised that scientific method. Cool. So learning intention then for today is we are going to define the key parts of the cell. and we will also talk about the types of transport. If you guys remember when I talked about the scientific method and the scope of the assessment, one of the things you're going to need to do is explain your results, and when you explain your results, you need to tie it back into the scientific theory. And so today I'm going to be covering some of that scientific theory so that way you guys can make good predictions and discuss your results and things like that. Sound good? So this is kind of looking at that material. Cool. And then last thing before we start getting to our learning is our weekly Fakatoki. So this week we have Hote Mori He Mea Huna Ki Te Moana, which is the life force is hidden in the sea. Powerful aspects are hidden in plain light. I was thinking about that when I was watching the news this morning and how they were talking about climate change and that need for us to make some real drastic changes in the next decade. And I thought a lot about the life force of the Earth. In particular, you can think about common things like the sea and how if we don't get on top of climate change, it's going to be a real problem. And we see it and we deal with it every day, but we often don't think about it. So think about your planet a little bit today and what we can do to take care of it. All right, lesson plan for today. The first thing we're going to do is consider the following question just to kind of introduce the idea of cells. I'll talk about the key components of it. We'll start looking at the membrane because that's going to be very important in understanding the experiment that you guys will be carrying out. Then we will do a little mind map. I think mind maps are really great ways, especially in the senior school, to think about ideas. If you're trying to get to that merit excellence level, you're going to need to show linkage of ideas, and you need to show that extended abstract thinking, that big picture thinking. Mind maps are a really good way to organize your ideas, so I highly recommend them as a means to revise and to make your own notes, and I will model that today when we are doing the transport. And then we have some Scipad pages to introduce what we've talked about and practice it. Cool? All right, so here's our success criteria. I will hopefully have a checklist for you by the end of this week. What we are looking at are the key parts of the cell today. So you should be able to identify the ones that we need. And you should also be able to compare the different types of cellular transport. We will work on this one over a couple of lessons. So you might not be able to fully compare and contrast those different types of cellular transport at the end of this lesson, but you should hopefully be able to define and identify which ones are available. Cool? All right, let's then get started with our consider the following question. So before I start giving you some notes on the cell, I want you guys to discuss in your groups some ideas. The first one is, why do you think our body needs skin? What's the whole purpose of skin in the role of our human body as an organism? And in fact, a lot of animals have skin. And then the second thing is, think about analogies. So I specifically asked about the human body and the skin. How do you think that can tie possibly to the cell? So take a think about those two questions, discuss with each other, and then we will get started. Why do we need skin? How does that relate to the cell? Cool. Let's get started. So, thinking about the skin, what does that skin provide us?
Warmth.
Warmth, yeah, it's a good regulator.
Protection.
Protection, that's a really good word.
It keeps all of our insides inside.
Yeah, I love that. Keeps all of our insides on the inside, which is all really important. The reason why I want us to think about the skin and the body and what it does is because we will use that as an analogy for when we're talking about the cells. In particular, one of our big focuses is going to be the cell membrane for this internal, and the cell membrane acts very similarly to the skin as a protection barrier and also as a regulator. Because we want to make sure our internal environment inside our body is a certain way, and that's going to be different from our external environment. Yeah? So, what we will do now is I will swap to the document camera. And I'll pass out this printout of the cell so you guys have a copy. And I will write some notes on there. Now, if you want the image, it is posted on my Google site. So you're welcome to copy and paste it into your online notes. Or you can find your own image of a cell. They all serve the same idea. Thinking about cells, so. So personally, I like to write on paper. Just my own preference. Yeah, I'm sorry that the text is really small. I figured I wanted a text so that way you guys do have the labels for all the different organelles, but we don't need to know all of them for this standard. So I just wanted to kind of use it as a means to kind of highlight certain sections. Cool. So let's swap to that, some of the key things in that document. Now, like I said, I apologize that the image is quite small, but I liked it because it gives you both a plant and an animal cell, which is good for comparison. The other reason why I like it is because it labels a lot of the organelles, and it gives you the function for those organelles as well. But I'm only going to highlight a couple of them that are relevant for us for this topic. So yeah. So first thing that I need to kind of talk about are what are these little lines here and what are organelles. So I'll make a note on what organelles are. I'm just going to zoom out a bit. So organelles are the components of a cell. And they have a specific function. And I'll just move that so you guys can have some space on there. So what I've done with this image is, like I said, they have provided a list of the organelles, and they've given a little bit of a description. I'm sorry, it's really tiny. You're going to have to squint to read it. But it's useful just to kind of know what is there. There's only a couple that you guys need to worry about for genetic variation and for this standard as well. So I'll just point them out to you. What else did I want to say about the organelles? Just trying to think. Yes, an analogy. So organelles to a cell is like organs to our body. So in our human body, we have multiple organs like the heart, the lungs, the stomach, and they all have a specific role to keep us alive. The cell is exactly the same. All of these organelles have a particular role to make sure our cells function. And obviously we are made up of cells. They're all working together as a team to keep us alive. Now, we already know one of these organelles that you guys have seen for genetic variation, and that is that nucleus, which is that big dot here. You can see it both in the plant cell and in the animal cell. And that's really important because it holds all of our genetic material. It basically tells the cell how to function and do its job. So that's why we need our DNA there. The one that we are kind of concerned for this topic is, let me point out a couple things that we are going to be talking about in particular. The first one that is going to be a key thing that we talk about is the cytoplasm. So I'm just going to circle that in blue, and I'll make some extra notes on the cytoplasm. So cytoplasm, it's basically the fluid that's inside of the cell. So when we're thinking about that inside fluid of the cell, it's obviously going to have all the different organelles kind of floating in it, but it's also going to have things dissolved into it, such as sugars and salts, water, those sort of things. So it's really important to think about what that cytoplasm is on the inside, because we want to regulate it from the outside. We want this inside environment to be different from the outside environment. And so we need to know what the cytoplasm is and what is inside it so we can compare it against what's in the extracellular environment. So basically anything outside of my cell membrane we call the extracellular environment. Shall I write that down? Extracellular environment is just the outside stuff of the cell. Outside fluid of the cell, or maybe liquid. Yeah, sorry about that. Yeah, extracellular environment. I should also capitalize the E and model good sentence structures. So the next question is, do you think the inside of the cell and the outside of the cell are different? Yes. So I need to regulate it, and the way that we regulate the inside and the outside of the cell is through that cell membrane. There's a delay. There we go. So the other important thing that we need to know for this topic is the cell membrane. That is going to act as our barrier to the inside of the cell, and it's going to control what comes in and out. So slightly different from our skin. So our skin has very limited functions in a sense of letting things in and out. It does absorb a little bit, and it does also release things like sweat. It's not as versatile as a cell membrane, but the whole idea is that it's in a barrier, so that way we can control that inner environment. Are we good with that so far? All right. The other thing to maybe kind of keep an eye out is when we have plant cells versus animal cells. So animal cells and plant cells both have a cell membrane, but plant cells do have a cell wall on top of having a cell membrane, so it has an extra layer. That cell wall is there to help give it structure. So we have bones and things like that, but if I have a plant, it needs to have structure so that way it can stand up. And so that cell wall helps it keep that rigidity for plants. Have you ever noticed a plant kind of drooping over, but it doesn't completely fall like flat? And the reason why it doesn't completely fall flat is because of the cell walls that help keep some of that structure there. The other thing that's different, I won't write that down because I don't think it's too important right now. The other thing to keep in mind that's different between plant and animal cells is that plant cells have a large vacuole. So they have basically this huge storage inside the cell. Animal cells do have vacuoles, but they're very small. So I've seen in some diagrams them labeled, in other diagrams they're not. But with the plant cells, these vacuoles are important to store water. It also stores nutrients. So when you think about plants and their ability to kind of survive droughts and things, the reason why they're able to do that is because of that vacuole storing their stuff. So when a plant is nice and, upright and not drooping over, it has a nice full vacuole with lots of water. As we go into droughts, it loses that water, and so that vacuole shrinks, and the plant cell will also shrink. And that's why we see it drooping over. But the plant cell doesn't completely droop over because we have that cell wall to help keep it up. The reason why I'm talking about this is because some of the practicals that we're going to do are going to involve plant cells instead of animal cells. And so we need to factor that in as part of our reasoning to what's happening and going on. Are we good so far with that? Questions? All right. Let's now talk specifically about the cell membranes. I'm going to go back into orange. And what I'm going to do is just like a little cross section of the cell membrane so that way we can have some notes about what we're looking at. So with the cell membrane, there are two layers. So I'm just going to draw a line of circles and another line of circles and some little tails. So, and I'm going to label that as the structure of the cell membrane. So, whoops, let me move that over. Actually, I might zoom that out. So, some things to keep in mind about the cell membrane. The first thing that's kind of a big overarching idea about the cell membrane is that it is semi-permeable. The other word you might hear it being referred to is selectively. Any ideas of what semi-permeable means? Vocabulary-wise? What does permeable mean? If I cut my skin, my inside is permeable. Bleeding is a good guess. It means that we can access it, right? So semi-permeable means that only some things are allowed through the membrane. Say, for example, things that are small. Molecules like water. I'll move that over. Are we good so far? Why would I want to be selectively permeable and not fully permeable?
Because if you let everything in, you let the bad stuff.
Exactly. If I let everything in, then I haven't actually managed and controlled my inner environment for that cell, right? And the whole purpose of me trying to build a wall is so that way I can manage what comes in and what comes out. Yeah? All right. So some things to kind of note about the cell membrane is that there's two parts to it. I'm just trying to grab some more color so that way we can kind of hit the whole rainbow. So what you'll notice is that I have these little circles and I have these little lines. The circles, actually I should say the whole thing is known as, and now I gotta remember the terms. I can't remember the term. I'll give you the term tomorrow. All right, so with these cell membranes, what you'll notice is that there's these little circles. Those circles are representing a hydrophilic side. Any ideas of what hydrophilic means?
Something to do with water.
Something to do with water, because hydro and philic.
Like water.
Yes. So this is a water-loving side. Does it make sense that I want the outside barrier and the inside barrier to be water lovers? Yeah, because this is mostly made out of water. That's mostly made out of water. So I want that to be a hydrophilic side. Then these little tails are hydrophobic. And that's how I'm able to control what's going in and out of the cell, because this stuff here is hydrophobic, that's hydrophilic, so only a couple things can go through it. So this section here is hydrophobic. And you guys probably have heard the word hydrophobic before. Some people might be hydrophobic in this class. I'm not a huge fan of water, but I don't think I'm hydrophobic. So hydro, water, and then phobic, to be fearful of. So it doesn't like water. Are we good with that so far? All right. The last thing that I want to kind of point out, now I'm trying to think about what color I haven't used. I haven't used green. So obviously when I think about that membrane, I do want to have things come in and out, but sometimes there's things that I want to get through that can't just go through the membrane. So what we have are protein channels, and I'll do that in green, and then I'm going to draw some more of the membrane. And don't worry, we'll be done with this section of notes. We have a protein channel. And that protein channel can allow things to get through the membrane. Maybe it's a specific molecule that I want. Maybe I'm trying to move things across a concentration barrier. But basically what this is functioning as is just like a normal tunnel that you'd expect to get through a mountain. Cool. All right. I am going to now move on to the next thing. Has everyone got what they need written down? No? Should I leave it on the board for a little longer? Okay. Is that good? Do you want more the other things? Is that all you need? All right. I'll just freeze that. And pause the recording. Cool. So, it's such a critical part. There is the PBS Learning. I'll show you where it is. So, go to Google Classroom, go to today's lesson, and there is this link to Cell Mem. And I want you guys to open up that link. And I want you guys to have a chance just to explore that cell membrane. Have a look at the different types of protein channels. Have a look at, what's inside the cell versus what's in the intestinal fluid. So this is like the intestine versus the inside of the cell. And also have a look at the different substances and how they move. So you can click on them and you can see how things are moving through that cell membrane. And there's also some paragraph as well to give you guys some information. I want you guys just to think about what those different proteins are used for, and also just identify which molecules can move through the membrane and which ones have to go through a protein channel. So have a couple minutes to take a look at that. All right, cool. Can I talk first and then give you guys some choices?
Yeah.
Okay, so this website is a really good way to visualize what we're talking about with the cell membrane and also that management of that internal and external environment. And like I said, you can click the different molecules and you can see which ones can go through the membranes and which ones can't, and which ones might rely on some of the proteins. Don't worry about writing down all that detail. I'm happy if you guys do want to take notes on it later, but just do it in your own time. I will talk about this in more detail when we start looking at the practicals and explaining how things are moving and why. The key thing we're going to be looking at for this internal is going to be diffusion, but diffusion of water, which is known as osmosis. So that water movement is going to be really important for this standard. If I click and my water can move. Okay, well, the animation is not working. Come on, water. Anyway, not working. Feel free to kind of bookmark that tab because it's useful for later when you guys are writing your report and trying to explain why things are moving across that membrane. Are we good so far? Yeah. All right. Now, I want to give you guys some choices because I don't want to sit and talk for too long. So there were two things left on our agenda, and that was to talk about some of the different types of transport. Now, I'm happy to give you guys the notes and explain it to you, or if you guys want, you can have a chance to do some book work because I know I've been talking for quite a while. You'll still gain the same information. So do you guys want me to give you notes on transport, or do you guys want to have a chance to do the transport on your own and do some Scipad work? Scipad work? Okay. So I'll skip the mind map. What I will do is I'll write up a mind map and I'll put it online if you guys want an example of one. So you guys can work on the Scipad. 25 minutes now. There are three things I want you to look at in that Scipad. The first thing is about the plasma membrane. That's on page 14. The second thing is an introduction to transport. That is going to be the new material. And that is looking at active and passive transport. So, with this one here, it's really important that you read that paragraph in the beginning so that way you know those different examples. And then the last one is concentration gradient. Trying to think. We'll probably pick up some of this material in the next lesson, so don't stress too much. I will walk you through and kind of explain it. Alright? So, go ahead and get some work done. You have about 20 minutes now. You are welcome to listen to music, but you need to have headphones, and you need to be listening through your device, not your phone. Okay? So, if you don't have your, and lastly, if you don't have your Scipad today, it is on Google Classroom, and you can edit it through Kmart. Call me over when you have work done, so I can give some feedback, and I can take credit and write down notes, and give you evidence for getting work done, and your stickers as well. I think this work only really goes up to the achieve level because it's just like foundational stuff, so just FYI. Cool. All right. Let me recap today's lesson and unfreeze it. That's not frozen. Okay. If you haven't finished the work for today, wait, listen up. I'm going to summarize. So, in our next lesson, I'll start talking about transport. So if you want, you can hold off on doing these 2 pages. But you should definitely get page 14 done in that Scipad because it recaps what you've done for today. So, to recap, you guys should be able to define the key parts of the cell, and you should also start to be able to hold up some of that transport that goes through it. Number 3, you should be able to do. Number 4 is a work in progress, so just keep that in mind. Great work today. Let me give you guys some coffee beans. What else did I want to say? Make sure you spray and wipe down my tables.
Did you know that?